will follow with the presentation of Peter Harrison. So I'm not Peter Harrison, but his plane was cancelled today, so he can only join tomorrow. So we have to do the presentation today. So it's more focused on the way we are producing functional annotation. So it's more on bioinformatics and data management. So the first aim of GENE-SWitCH, again, I will repeat it a bit, but it's the same picture, so I won't detail much, is to create a freely, so it's really open access, high-quality maps of the function of pig and chicken genomes. So I think you're aware of the ENCODE project for human. It's the same, with a bit less ambitious, but it's the same for a livestock species that we are trying to do at FONG. And in our case, really to focus on pig and chicken. So the idea is to annotate a wide range of tissue, and not only tissues, but also developmental stages. And because we do that, because we really think that it underpins the phenotypes of adults. So by knowing things, I will get it now. No. Okay, anyway. By getting knowledge on the early stages of development, we think that will be better to explain the phenotypes in adults or young animals. And so we also include specific translational studies, but I won't come back because it's the same as I said before. So now, just getting more in details, this project is part of Europhong. So Europhong is a coordinated effort across 5 large European projects, now 6, I think, because we added holoruminant yesterday, to standardize our research processes, analysis, and pipelines and methods. So we decided that all these projects on the right, so GENE-SWitCH, RumiGene, GEroNIMO, BovReg, Aquafong, and holoruminants, will try to manage the same way the data, the production of data, the storage of data, the accessibility of data. And this will increase effectiveness at collectively improving animal productions, welfare, and supporting industry applications. So this is part of FONG. So FONG is a functional animal annotation initiative to really improve the annotation of livestock species and companion animals too. And the idea is really to use this knowledge to improve our predictive power through the combination of standardized data. So what we have now with GENE-SWitCH, so we just finished the production of data. So we have sequenced around 3,000 samples from 350 individuals. We have improved and we have mostly standardized the sampling and the analysis of all these data sets for the 2 species. And for that, we also have open access bioinformatic analysis methods that are open to everyone, such as the Tagada pipelines, which is able to do an automatic annotations of a tissue from RNA-seq data. And by the way, we are also working on the importance of this information to understand better the traits, the building of phenotypes. So this information can be accessible through the Ensemble genome browsers. I think most of you know what it is. It's an internet databases that is funded by Europe, and which allows to aggregate sequencing informations. It includes the majority of major animals for agricultures, but also for crops, so vegetables, and aquacultures. And the site is updated 4 times a year with new data integrations. So every 2 weeks, you also have a rapid release to integrate all the data that have been uploaded during each day. So finally, for EBL, so I'm not really expert for this, so I let you see. But the idea is that everything is standardized. So starting from the reference genomes and the data, so long reads, RNA-seq, protein information, finally, you will get a model that will proceed to the annotation. And this is not open access, so the pipeline is not open to the public, but the way the annotations are produced by EBI is standardized, and it will be always the same. And at the end, you will get quality reference annotations. And by using the GENE-SWitCH dataset, it will include also developmental time points and tissues. And it will increase significantly what is today available on the Ensemble database. That if you're working with, you can know that it's not totally perfect. The idea is also to combine Ensemble data with all publicly available data to make the best possible annotation. So not only the GENE-SWitCH project, but all the things that can be made to improve the annotations for these two species. And you can easily download the new annotations from the EBI website. So now, for the GENE-SWitCH project, all the data and analysis we have produced are accessible on the Ensemble web page, but so on this direction, email address. No, on this internet address. And what you can see also, so you can get many different information. You can get GTF, so annotation files. You can get FASTQ files. You can get also BAM files, bed files, depending on the data we are processing. And so this is all available on this internet place. And what we are also trying to develop is a way to see the data. So when you want to look at a specific tissue gene expression and transcript data expressed in a specific tissue, you can get this type of windows where you can see the different isoforms we have identified within the project. So each year for GENE-SWitCH, we are contributing to the regulatory builds from EBI. And the regulatory build is really is not based on transcripts, but it is based on functional information we can get to really say that in the genome, for example, the regions that are in red here are probably regulatory regions. So you have on one side the genes and the transcripts, and on the other side you have the regulation. So how genes are regulated to be expressed or not in the different tissues we are working with. So now it's not always easy to access this information and to know how to get it. So within GENE-SWitCH and in collaboration with EBI and other partners like EFAB and EAAP, and within the GENE-SWitCH project, we are a dedicated outreach and training team. And we are doing videos and tutorials to explain how to use it. You have also a support help desk to help you if you have any questions. So do not hesitate to connect and to use it. Moreover, we are also providing trainings. So the next one will be the 13th of January next year. It's a 2-day technical course, and it's in Ixton, close to Cambridge, on the EMBL EBI campus. And the idea is to help stakeholders to use the data that we are producing now with GENE-SWitCH, and to use it for genomic predictions. Also within the models we are developing, but perhaps for other applications too. And to get a strong support to be able to use this information on Ensemble to exploit pig and chicken genes, variation, and regulatory annotations. So please do not forget to, if you are interested in to save this date, it's free for people who apply, and it's free. Yeah, to participate is free. I'm not sure it's on site or not, I don't know. So you can also contact Peter Harrison if you want more information on this training. So finally, what we can say is we are trying by GENE-SWitCH to discover new traits of interest, a new way to understand how traits are built. GENE-SWitCH data leveraged through the Ensemble genome browsers provide a powerful environment for exploring traits of interest in genomic and regulatory context. GENE-SWitCH data will be continued and will be interrogated by GENE-SWitCH scientists, but also other scientists, to identify novel traits of interest and improve predictions. We are still improving analysis pipelines, and these new pipelines will be available on the GENE-SWitCH website and on the Functional data portal. The updated reference for pig and chicken annotation are of huge value to the community, and Ensemble will continue to update them as the future data becomes available. So it will not be finished at the end of the project, but we have a follow-up of this. And an important point is that the new regulatory bills will represent a major advance for agricultural genomics. So we are really, within the project, trying to improve this part of the functional annotations to be able to really enrich the prediction model with this type of information, at least to test if it's relevant or not. Moreover, we have and we will have in-person and virtual training resources to assist scientists and stakeholders, like industry experts, to exploit at the maximum the GENE-SWitCH data that are available in Ensemble. So thank you for your attention, and I'm a bit sorry because I'm not really aware of all the things that are inside the presentation. But I think the main message is there, so thank you for your attention.